Now to discuss some of the studies being presented in mantle cell lymphoma and T-cell lymphoma is Dr. Owen A. O'Connor, who is the American Cancer Society Research Professor, Director of the Center for Lymphoid Malignancies, and Co-Program Director of the Lymphoid Development and Malignancy Program in the Herbert Irving Comprehensive Cancer Center at Columbia University Medical Center. Hi, Dr. O'Connor. We're so happy to have you today.
Thank you. Thank you, Gina.
Sure. So let's first talk about what's going on in mantle cell lymphoma at this year's meeting. What are some of the things that you would like to mention?
I think there's little question that the BTK inhibitors continue to evolve and are a class of drugs that Michael Wang first reported on in the New England Journal of Medicine that demonstrated pretty remarkable activity. In Michael's earlier work, he showed an overall response rate of about 65% with a CR rate of about 20% with ibrutinib. As everybody on the planet knows by now, ibrutinib is a irreversible inhibitor of BTK and has demonstrated also very meaningful durations of benefit. On the heels of ibrutinib, a second generation BTK inhibitor, acalabrutinib, came along. Acalabrutinib is best distinguished from ibrutinib because it is felt to be a little bit more selective on the kinome compared to ibrutinib. And most importantly, it doesn't hit TEC, one of these enzymes that people have attributed the risk of bleeding to. Acalabrutinib was also active, but I think at this meeting, one of the things that's new is some of the long-term follow-up data for ibrutinib and acalabrutinib, which have continued to confirm very good durability of response and really no change in much of that toxicity profile that was originally reported. But there's a third generation BTK inhibitor, another covalently bound BTK inhibitor called zanubrutinib being developed by BeiGene. And so there is some interesting data with that drug. And that drug again is purportedly to be more selective against the relevant kinases of the kinome, not hitting TEC and some of those others that people attribute the toxicity to. But in some data presented at this meeting, it demonstrated a pretty high overall response rate of about 90% or so, 80-90%, with a CR rate of about 20%. And while that in and of itself seems interesting, the side effect profile for all the world looks like ibrutinib and acalabrutinib. There was really interesting data presented by Dr. Song from Peking University. And they did a study in China with about 84 patients, so a much larger single-arm kind of study. And they reported in a more heavily treated population than the first experience in the US, they reported an overall response rate of about 84-85%. But what sort of stunned people was the complete response rate. And the complete response rate in that Chinese population was about 60%.
Wow.
So if you go back and you look at Michael's data from acalabrutinib and ibrutinib, those floated around 20%. And if you look at the Chinese data, it's nearly tripling that CR rate. And as you might imagine, the expectation is that by having more CRs or deeper complete responses, you're going to get much better progression-free survival curves and much better durations of response. So the big question is why? Why is it that the Chinese experience has a much better complete response rate? And there's lots of reasons. You know, people have talked about likely issues revolving around cytochrome P450 and metabolism and some of these important ethnopharmacologic determinants, but I think it remains to be seen. We didn't see a lot in terms of PK. We didn't see a lot in terms of kinome scans and other things. It remains an interesting observations. These are small studies, so it could be a fluke, but it's definitely an interesting scientific piece of information to follow up on.
Yeah, it is interesting, definitely. Yeah. Now, honing in a little bit more on acalabrutinib, we were seeing some more data with it as a single agent as well as in combination. Do you want to go into those data a little bit more?
Yeah. So there was interesting data presented looking at acalabrutinib plus BR. And as most people are aware, there's been a massive shift, thanks in part to Matthias Rummel's data, where he demonstrated that BR seemed to be safer and as good as R-CHOP-based chemotherapy regimens in mantle cell lymphoma. So easier regimen. I don't know if it's a standard of care, but it's probably pretty close. So given the activity of the BTK inhibitors, it makes complete sense to try and deepen those responses. Now, some might look at this and say, you're just making soup adding all these drugs to a standard like BR. But in fact, there's probably a good rationale, because in mantle cell lymphoma, achieving PCR negativity is probably important. And there's really not a lot of data in the context of these kinds of regimens and their ability to do that. We know from this meeting this year and last year that the ability to achieve PCR negativity in CLL is probably very important in terms of its association with long-term survival. And we know from the Nordic Lymphoma Study Group that those patients who are PCR negative for the 11-14 translocation do better. And if you have a relapse of the PCR evident disease, you can treat with Rituxan and they still do well. So we didn't see data on PCR monitoring in this study, but they looked at a hodgepodge of patients. Patients that had relapsed refractory disease, as well as patients that were treatment naive. And as you might expect, the response rates in the treatment naive patients were really good. Overall response rates in the 90% range and CR rates in the 70% range. And in the relapsed refractory population, still very good. Overall response rates of about 84%, CRs probably in the rate of 50%. So that represents a pretty promising regimen, and I think it does raise the possibility that it could be explored in a randomized phase 3 clinical trial of BR against the acalabrutinib BR. But I would like to see some PCR testing and see if this is an important issue, and see if in fact the addition of a BTK helps us by achieving greater levels of not just complete remission, but PCR negative complete remissions.
Yeah, very good points. Now, we're also seeing some long-term data with a BCL2 inhibitor, venetoclax. So what are we seeing with this agent now, and what does that tell us about the role that venetoclax has in the MCL paradigm?
Yeah, you know, Matt Davids has presented a lot of this data, and you know, as one of the guys that helped develop bortezomib 100 years ago, and you know, that was the drug, it's really interesting to see now all these drugs. And you know, Constantine Tam has begun looking at BTK inhibitors plus venetoclax, and you might imagine that just like in CLL, where we're really not so much talking about FCR or bendamustine in CLL anymore, people are pretty optimistic that we're going to achieve pretty durable complete remissions in patients getting combinations of venetoclax with BTK plus CD20. The same paradigm is evolving in mantle cell. Now, mantle cell is a little bit more complicated because it is a broad spectrum of disease. Most of these studies have been happening in patients with Ki67 indices that are low. And so in mantle cell lymphoma, the Ki67 index, that measure of how proliferative the disease is probably the most important prognostic feature of the disease. It might correlate with the 17p11q in CLL. And so there's not a great description of what that patient population looks like in these studies, and I would like to see more of that as we go forward. But most of the studies are done in patients with more indolent mantle cell lymphoma. And so that's the case with venetoclax. And so what we know is that these combinations work well together. We're not really addressing how well they're working in the different risk populations, i.e. those that might have more advanced high proliferative disease versus those with lower proliferative disease. But venetoclax is yet another drug that's producing extremely high response rates. Back in the day with bortezomib, when we got 30% and 6 months everybody was ecstatic. Now that's old news and probably wouldn't pass muster today. Many of these drugs are hitting overall response rates of 70 to 80% with CR rates in the 54% range, and progression-free survivals and durations of response measured in years. So the paradigm is changing, and as you can see, every year we talk about this, it's changing fast.
A lot of potential going on.
Right.
Yes. So let's move on now to T-cell lymphoma.
Yeah.
What is going on in the frontline setting?
Yeah, you know, finally that disease that we as a medical community had neglected for quite a long time, or been addicted to using the treatments in B-cell lymphoma as the standards, things are finally changing. And at this meeting, there were two interesting frontline studies. One was the Echelon 2 study. And the Echelon 2 study is a very interesting trial. It looked at adding brentuximab vedotin, which is a CD30 targeted antibody drug conjugate, to the CHOP backbone. So it was a randomized study, largest to date, looking at CHOP against cyclophosphamide, doxorubicin, brentuximab, and prednisone. So the trial, interestingly, required that the eligible population, be made up of 75% patients with anaplastic large cell lymphoma. Now that, of course, is the disease where brentuximab got its early approval, and it's a disease characterized by 100% expression of CD30. The rest of the other PTCL subtypes don't express CD30 to the same robust extent. And so the response rates for BV and ALCL led to its very rapid approval. So in this trial, 75% of the patients had ALCL, and only 25% had non-ALCL. So the naive observer might say, oh, you stacked the cards in your favor here by putting in the subtype with the greatest vulnerability to that drug. But in fact, that 75% was done for a regulatory purpose. It was in response to the EMA, who required a post-marketing randomized study with the drug to demonstrate to confirm its activity and regulatory approval. So while there's no denying that 3/4 of the population had ALCL and they're more sensitive, the study was positive for both progression-free survival and overall survival. So the first time we've ever seen that. Now the skeptics are going to say, what about the other subtypes? How much CD30 expression do I need to treat those patients? And it's interesting that the trial required patients to have 10% CD30 expression to get in. But when the FDA approved the regimen, approved only 11 days after the submission, so a world record.
Wow.
The FDA approved no level of CD30, just CD30 positive. And so it raises interesting questions. Is CD30 important in the predicting the response or associating response? But I think the data are the data. If you have a CD30 positive T-cell lymphoma, this ACE, Cytoxan, doxorubicin, BV, prednisone regimen is the new standard of care. Another study, not positive, is ACT-1. And ACT-1 is presented by Francesco D'Amore from the Nordic Lymphoma Study Group, and looked at adding Campath, targeting CD52, to CHOP-based chemo. So they did a very good study looking at CHOP against Campath CHOP consolidated by stem cell transplant. And so that study in the beginning was wrought with all sorts of challenges, including CMV, viremia, and infections like fungal infections due to the anti-CD52. They made the appropriate dose adjustments and a lot of that toxicity went away, but the final results presented at this meeting show no difference in progression-free survival or overall survival for those two arms. So good news, bad news. At the end of the day, we've made an incremental step forward with Echelon 2.
Yes, absolutely. And now, overall, what are some other abstracts in non-Hodgkin lymphoma being presented at this meeting that you're excited about?
Well, staying on the theme of T-cell lymphoma, I think that there's a lot of exciting stuff happening. And Lorenzo Fauci from my team is going to share some data looking at epigenetic combinations in the disease, where we're now using these as frontline therapy. And we're seeing response rates in the 80 to 90% range, and some subtypes where 8 out of 9 patients are responding. So looking at combinations like azacitidine and romidepsin and some of these novel drug combinations in T-cell lymphoma are beginning to get some land legs, and they're beginning to evolve in a pretty favorable way. We heard earlier yesterday about the Augment study, and the Augment study was also a study I was involved in on the DSM committee for many years, and it looked to randomize in patients that had one line of prior therapy with follicular or marginal zone lymphoma, Rituxan versus Rituxan plus lenalidomide. And the study was presented yesterday and showed a substantial improvement in the progression-free survival. So now that was also a very positive study suggesting that adding lenalidomide to rituximab really produced, and it wasn't a trivial, small difference. It was a really, what I feel and most others believe, is a clinically meaningful improvement in the progression-free survival for patients with follicular and marginal zone lymphoma.
Oh. Well, Dr. O'Connor, this was wonderful. Thank you so much for sitting down with us and highlighting all these great abstracts.
You're
Appreciate
welcome.
it.
Thank you.
Great, thanks.